Hello everyone. This topic is about thin layer chromatography. Definition: thin layer chromatography is a technique used to isolate non volatile mixtures. The experiment is conducted on a sheet of aluminum foil, plastic, or glass, which is coated within a thin layer of absorbent material. The materials usually used is aluminum oxide, cellulose, or silica gel. Principle: thin layer chromatography depends on the separation principle. The components in the mobile phase moves over the surface of the stationary phase. The movement occurs in such a way that the components which have a higher affinity to the stationary phase moves slowly while the other components travel fast. The separation of the mixture is attained. Thin layer chromatography is widely used because of simplicity, relative low cost, high sensitivity, speed of separation. This is the picture of TLC. The beaker inside has TLC plate. TLC plate has solvent font sample spot and under the beaker has solvent. Now see the components of thin layer chromatography. Thin layer chromatography plates, preferably ready-made with a stationary phase. These are stable and chemically inert plates. The stationary phase on the plates is of uniform thickness and it is a fine particle size. TLC chamber. This is used for the development of thin layer chromatography plates. Chambers maintains a uniform environment inside for proper development of spots. It also prevents the evaporation of solvents and keeps the process dust free. Mobile phase. This compresses of a solvent or solvent mixture. The mobile phase used should be particulate free and of the highest purity for proper development of thin layer chromatography spot. Chemically inert with the sample, a stationary phase. A filter paper. This is moistened in the mobile phase to be placed inside the chamber. This helps develop a uniform rise in a mobile phase over the length of the stationary phase. This picture shows the stationary phase and mobile phase. Stationary phase as sample spotted on TLC plates. TLC plates are transferred to a TLC chamber. The mobile phase has components separated based on their polarity over time. It has compound A, compound B, compound C, and original sample. Procedure: to apply sample spot, thin marks are made at the bottom of the plate with the help of a pencil. Apply sample solution to the marked spots. Pour the mobile phase into the TLC chamber and to maintain equal humidity, place a moistened filter paper in the mobile phase. Place the plates in the TLC chamber and close it with a lid. It is kept in such a way that the sample faces the mobile phase. Immerse the plates for development. Remember to keep the sample spots will above the level of the mobile phase. Do not immerse it in the solvent. Wait until the development of spots. Once the spots are developed, take out the plates and dry them. The sample spots can be observed under a UV light chambers. RF value. On complication of the separation, each component appears as spots separated vertically. Each spot has a retention factor. RF equal to distance traveled by sample divided by distance traveled by solvent. The factors affecting retardation factors are the solvent system, amount of material spotted, absorbent and temperature. Thin layer chromatography is one of the fastest, least expensive, simplest and easiest chromatography technique. Applications: the qualitative test of various medicines such as sedatives, local anesthetics, anticoagulant, analgenesis, antihistamines, steroids, hypotonics is done by thin layer chromatography. TLC is extremely useful in biochemical analysis such as separation or isolation of biochemical metabolites from its blood, plasma, urine, blood fluids, serum, etc. Thin layer chromatography can be used to identify natural products like essential oils or volatile oils, fixed oil, glucosides, waxes, alkaloids, etc. And it is widely used in separating multi-components pharmaceutical formulations. It is used for the purification of samples and direct comparison is done between the sample and the authentic sample. It is used in the food industry to separate and identify colors, sweetness agents, and preservatives. It is used in the cosmetic industry and it is used to study if a reaction is complete. Disadvantages: thin layer chromatography plates do not have longer stationary phase. The length of separation is limited. The results generated from thin layer chromatography are difficult to reproduce. Since thin layer chromatography operates as an open system, some factors such as humidity and temperature can be affected the final outcome of the chromatogram. The detection limits is high and therefore if you want a lower detection limit you cannot use thin layer chromatography. It is only a qualitative analysis technique use and not quantitative. Advantages: simple process of component separation. Fewer types of equipment are used. Separation is achieved in a very short time. Components of UV lights are achievable to visible. Cheaper chromatographic technique use. And that's all about the thin layer chromatography. Thank you.